डोंट इरिटेट एनीवन। लास्ट मोमेंट पे किसी को कुछ नहीं करना है और किसी को अटैच नहीं करना सिस्टम ठीक है।
జూమ్ బంద్ చేసినావా? జూమ్ బంద్ చేసినావా? జూమ్ బంద్ చేసినావా?
अभी स्ट्रीम किया स्लाइड स्ट्रीम कर। हाँ आ रहा है ना। नो शेयर स्लाइड मिनिमाइज।
शेयर कर रहा हूँ 1 मिनट। हुआ?
नहीं आ रहा ना।
आ रहा है सर, देखिए कमल लोग सर लाइव जोड़ो।
आ गया ठीक है। लीव इट।
एक मिनट माइक कैमरा ऑन। चलिए इट्स फाइन। नहीं लेते हैं। That this will work, that will not work.
That will not work. We'll give this option. Yatish.
Suggesting we should on the mark the lighter is highlighted.
Oh that is big.
इधर वो स्लाइड में नहीं दिखता ना, इसको नहीं।
And slide thing changes course. ఇది కూడా వచ్చేది
పంజెట్లో
ఇది పనిచేయట్లేదు సార్. ఒకసారి పని చేసుకుంటే
Yatish, shall we start? Wait.
you can use this.
అది అంటే ఇక్కడ పాయింటర్ ఉంటుంది కదా,
కానీ పాయింటర్ దాని మీద రాదు, ఈ గ్రీన్ వస్తుంది. You can change slide from this.
पूछना पड़ेगा
मुरली इसके बाद में फ्लैश नोट्स, इसके बाद में आप पैनल है ना, मैं just then only will take care of panel. After panel discussion, we will be programs. To say the field validate on the first to share their opinion. Now we'll start one by one. Yatish, shall we or? Welcome back. We'll now start the session 2. Now, the next speaker is Dr. PK Sharma. He is GM and Head Technical in BIRAC, Biotechnology Industry Research Assistant Council. Dr. Sharma is a trained medical chemist with cross-functional research experience of over 15 years in pharma industry. He did his PhD from University of Hyderabad and postdoc from Queen's University, Belfast. And he specialized in medicinal chemistry, organic synthesis, methodology development, and process development. I would now invite Dr. Sharma to give his talk. Slides.
Slide change.
Yeah.
ये pointer है सर, ये pointer green.
ठीक है.
Good morning, all. I'm PK Sharma. I represent BIRAC, that is Biotechnology Industry Research Assistance Council, New Delhi. This is set up by Department of Biotechnology as a not-for-profit organization. And we support affordable product development and encourage public-private partnerships. So I thought I want to present here the opportunities available for PhDs and postdocs who wants to become an entrepreneur from BIRAC. What are the opportunities available? Briefly, I will present. I am not going to go through all the slides what are there. I will present what it is. Yeah. A typical product or technology development cycle starts from ideation, and then it goes to proof of concept. This one. And then early stage development and late stage development, and finally product commercialization. So there are schemes available from BIRAC for all these technology readiness levels. Actually, the product or technology development is determined in terms of technology readiness levels. Ideation means it starts with TRL 0, and commercialization means it ends with TRL 9. So for ideation, for example, any postdoc or PhD, if they want to become an entrepreneur, they can join our SPC centers, which are located at different incubators. So they can join there. They will be given a fellowship and contingency also. They can go to field and identify the problems in the field, and they can formulate their idea for development of product or technology. Once they come back with that idea, they can submit their proposal to BIG, that is Biotechnology Ignition Grant, where the funding will be given to develop proof of concept from idea. That is TRL 0 to TRL 3. 50 lakhs will be given for 18 months so that the individual can develop a proof of concept for their product or technology. An individual also can apply for this grant, but they have to be incubated with one of the incubator, or any startup also can apply for this grant. What we expect is by the end of BIG project, we expect them to start their own company if they submit the proposal as an individual. Once the proof of concept is generated, then they can submit the proposal for SBRI, SBIRI. That is for doing early stage validation. Here, what we do is like Timelines also, we have not specified anything. Depending on the requirement of the validation of the product or technology, they can indicate the timelines. Here the funding will be 50 lakhs will be given by BIRAC. Over and above, if the project cost is more than 50 lakhs, that should be shared between applicant and the BIRAC. And when idea to proof of concept, definitely nobody will be ready to fund the technology. So that is why government is giving 50 lakhs for that one. Once the proof of concept is there will be some investors to fund the project. That is why we have put that up to 50 lakhs we will be given as grant. They will be given as grant. After that, they have to share the money. If it is 60 lakhs, for example, 55 lakhs will be given by BIRAC and 5 lakhs has to be by applicant. Once the early stage validation is done, if they want to do the late stage validation, they can submit the proposal for BIPP, where the funding is not limited. The thing is, the product cost should be shared between applicant and the BIRAC. That is, if it is 100 lakhs, 50-50 they have to put. And once the validation is done, then they have to go for the product commercialization. There is product commercialization program fund. Once the validation is done, TRL 7 is completed. If they want to do the commercialization large scale scale up, they can submit the proposal for product commercialization program fund, where we will give to 5 crores funding. But the thing is, once the technology is successfully commercialized, they have to return 150%. If they take 5 crores, they have to give the money 7.5 crores, they have to return it to BIRAC. But it is in easy installments. In 8 years, they have to give that money back. Yeah. 8 years. 80, not 80 years. 8 years. But we are planning to revise the guidelines. That will be done. Sparsh centers are already located at different incubators. They can approach them. They will announce the call for identifying the Sparsh fellows. So they can apply there. And if they are selected, the mentoring will be provided by incubators and they can identify the problem. For big, the incubators again, they will give the mentorship and all. For SIBRI and BIPP, what we do is, once the project is funded, we assign 2 experts for each proposal so that they will be monitoring, not only monitoring, they will be mentoring the project also. So in addition to giving funding, we are providing mentorship also and incubation, of course. If you are submitting the proposal as an individual, if you don't have any facility, you can go to any of the incubator and start your work. So if you have an idea, good idea, and if you want to have funding, so you can start with, as an individual also, you can start your journey for the product development. Mentorship is there. And what we do is like regulatory support also we give. Once the project is funded, we identify the regulatory requirements at each milestone. What are the regulatory requirements? So that startup will know what to be achieved before completing a particular milestone, what are the regulatory requirements. And in addition to that, patent filing also can be done through BIRAC support. There is path scheme is there. Through that, they can file patents also. Just I will go to details of these schemes. This is startup ecosystem nurturing. There are several programs out there under BIRAC. I don't want to get into details. Thematic areas under biotechnology, we found all areas of biotechnology, that is drugs including drug delivery, biopharmaceuticals, vaccines, devices and diagnostics, industrial biotechnology, agriculture, aquaculture, bioinformatics, everything under biotechnology will be supported. And these are the incubators I was mentioning. There are 75 plus incubators given by BIRAC actually. So you can incubate in any of the incubators to take your product or technology forward. And there are UUA centers also. So these are set up at universities where these UGs or postgraduates and PhDs, they will be given mentorship to identify the product, the ideas. This is the BIRAC, BioNest, and UUA network. There are 75 plus incubation centers are there. EUA centers are also there. 25 EUA centers are also there. And this is the biotechnology ignition grant scheme I am talking about. The call will be announced twice a year, and 1st January and 1st July. Each innovator receives up to 50 lakhs for research projects for 8 months. And the scheme is currently managed through 8 big partners. So you have to submit your application to one of the big partner, and the proposals will be evaluated centrally, and finally they will be given. And this is what I'm talking about, CBRI and BIPP. Again, these two are twice a year, these calls are announced. So we can submit the proposal anytime, and funding will be given as mentioned earlier. And this is for academic researchers actually. If there is any academic researcher out there, if they want to take their product or technology, this is the AIR scheme. So this scheme promotes development of proof of concept. If you have some basic data, if you want to develop a proof of concept, we will give that academic innovation research fund we will give. That is for 50 lakhs for 24 months. The difference between big and this is, in big, you can start with idea, but here we expect some basic data from your lab. This is for academic researchers. Once you have the proof of concept, you can go to the contract research scheme. That is for validation of your technology. You can identify an industry for doing the validation work in a contract research mode. So IP will stay with the academia, but the money, whatever is required for validation, that will be given by BIRAC. So first right of refusal will have the CRO or whatever industry who will be doing that validation work. If they are not interested, you can go to other partners. This is for academic researchers. This is early translation accelerator. Again, it is for academic researchers. So we have set up these early translation accelerators at different places, like different incubators. For example, IIT Mumbai. And some of the incubators, CCAM, IIT Madras also, we have started this one. The idea is to take the academic discoveries into products. See, normally what happens, the publications are patents generated by academia will not be taken by industry as the translation component is missing in those discoveries. So what we do here is the early translation accelerators, they do the translational work, which is required for industry, and maybe 5 minutes more. They provide the translational component, which is required by industry, so that once the validation work is done for the academic discovery, the industry will take it forward. That is the idea of setting up of these early translation accelerators. Next, product commercialization program fund. I mentioned about this. Here the eligibility is validation should have been completed for their product or technology, and it should be a startup. That is the definition of startups, less than 10 years. If they meet that criteria, they can submit proposals to PCP fund. So far, we are taking proposals for this fund throughout the year. Now we are thinking of announcing a call twice a year that we are going to do and revise the guidelines. And I mentioned this one. There are for entrepreneurs, there are seed fund, leap fund, and AS fund also, where they take some equity and all. In addition to whatever funding is given by BIRAC, they can opt for these things also. These are also managed by BIRAC. And supporting partnering activity. See, the idea of showing all these things are there. These are all regular schemes, big CBRI, BIPP, PCP, all these things. In addition to, we will be announcing calls, one-time calls also, with some collaborations. So keep visiting our website. There are opportunities for funding. So this is Welcome Trust Program. This is one-time call. Cefipra, we have done something with them. And USAID we have done, and Nesta, IIPME with the Ministry of Electronics and Information Technology. We have done this program in the area of medical electronics. With the DBT also, we do some joint calls. So these calls also, if you see our website, there will be many opportunities. And our funding is always based on the technology readiness level, either TRL 3, 4, 5, 6, whatever it is. Wherever you fit, you can submit the proposal in any of these calls also. These are also one-time calls. We have started synthetic biology. To support this area, we have already done 2 calls, and we are planning to do one more call in the area of synthetic biology. Again, we are planning to support proof of concept generation and validation also in this call. And recently, preclinical models for drug discovery. We have announced a call last year, and we have supported some of the projects for the development of preclinical models. This is also one-time call, but mostly industries and even academia also submitted proposals for this. We are going to fund them. Mission COVID Suraksha, that is the integrated approach to vaccine-induced immunity. We address support vaccine-induced immunity. So we have announced this call, that is one-time call to address that vaccine-induced immunity. We have supported some proposals. These are all one-time calls. And Gaurgam, Secondary Agriculture Entrepreneur Network. In the state of Punjab, we have supported this network to encourage secondary agriculture particularly. That we have done with NABI, SIAB and Bionest PU, where Punjab State Council for Science and Technology, they have managed this program. As all of you know that during COVID times, we have announced many calls, we have supported projects. This is secondary agriculture. This is all our achievements. I don't want to go into details. This is Mulankar Bioenabler. Dr. Gokhale has mentioned about this one. The last date for this is July 24th. So if you are interested in setting up the facility, either green or brownfield facility or using the existing facility, you can submit the proposal for this one. This is under the biomanufacturing initiative. This is Global Bio, 12 to 14 September 24. It's going to be held in Pragati Maidan. Registration we are going to start. So it will be a good opportunity to interact with people and all. So this is all. As I mentioned, please keep visiting our website regularly and feel free to write to us anytime. So we will be responding. Thank you for this opportunity.
Thank you, Dr. Sharma, for giving talk about the BIRAC and its new projects. No, I request Dr. Bhandari. Momento. Dr. Sharma.
Now we will move on to the student flash talk. Can I invite a portion number from 21 to 25, please to come forward, please. Portion number 21 to 25.
Hi everyone, my name is Rameshwari. I'm from IVSC and working under the supervision of Dr. Nanaucha Sharma. And my research work is biochemical and molecular characterization of freshwater cyanobacteria from northeastern India for the potential to inhibit different forms of cancer propagations. And as we all know that the cancer chemo resistance is developing so fast nowadays, and in order to overcome those of this chemo resistance, we are aiming to focus more on the cyanobacterial derived compounds. And because of that, I have with this perspective, I have extracted carotenoid from Toliprotex extract, and this Toliprotex extract used for the synthesis of nanoparticle in zinc oxide and encapsulated with the cisplatin. And with this, we found that it will sensitize that oral cancer cell line, cisplatin oral cancer cell line. And another one is phycocyanin. We have purified phycocyanin from the Westilopsis species, and we got a purity of 2.3, and which is the purity index of phycocyanin used in the food and the cosmetic industry. And additionally, it has also an anti-cancer activity in MDA231 and through cisplatin chemo resistant caspase-3 independent pathway. apoptosis pathway. And the lastly, that is the dolipotric screw extrude column fraction, that is the TDF. In this TDF fraction, I have found that it will inhibit the cell proliferation and cell metastasis of the chemoresistant oral cancer cell line. And we also find out the mechanisms of the sensitizations, and it reveals that it will through that PI3K beta-catenin pathway. And I think it will be a very interesting to know in details. So please visit in the poster number 21 for more details. Thank you.
Good morning, another person here. I am Hima Parvati from Rajiv Gandhi Center for Biotechnology, working under the supervision of Dr. E.V. Sonia. So I am basically an agriculturist and a microbiologist, and now I am becoming a biotechnologist. So my approach is mainly farmer friendly, and I have to reach to the farmers. So the topic I have chosen for my PhD is the microbiome that is affecting the plant and the whole ecosystem. So mainly our work is mainly focused on the Piper diagram, which is black pepper, known as the black gold. So my area mainly focuses on the microbiome part, which is mainly the important part of an integrated management of pests and diseases. So we mainly focus on the disease caused by an oomycete pathogen, Phytophthora capsicum, which is a soil-borne disease. So we are mainly focusing on the mapping, and we are comparing the microbiome diversity of a resistant Piper species with the commercial Piper species. So with that, we are purposefully mapping the microbiome diversity. And finally, our output is mainly focusing on coming out with a microbial consortia, which is mainly in the resistant rhizobiome, which can help in recovering the susceptible plants from this particular disease. Thank you.
Good afternoon, everyone. My name is Surmani Huidrom. I'm from IBSD. I'm working under the supervision of Dr. Indira Devi. My PhD topic is on bioprospecting of lactic acid bacteria from fermented beverage for antimicrobial and probiotic properties. As we know, Northeast has various bioresources. Among is this fermented food. As we know that fermented food is the rich source of beneficial microorganisms like lactic acid bacteria. So in my objectives, I have taken fermented beverage, which is very common in Northeast India, which based on the traditional knowledge, they have been used for ethnomedicinal properties for various Lyme diseases. In my study, I have evaluated in vitro from fermented beverage isolated lactic acid bacteria. Most of the prominent were lactobacillus species. Among this lactobacillus species, I have carried out for antimicrobial and probiotic properties. Among this, we are few lactobacillus. Few has shown a very good potential against antimicrobial, against the escape pathogens, as well as a good promising candidate for probiotic this thing. So based among this lactobacillus strain, one of the strain which I have taken, which has a very good promising candidate for both antimicrobial and probiotic candidates. So based on genomic insight study, I have we have carried out the prediction in this genomic mining that there is a potential this genome of Lactobacillus plantarum has harboring biosynthetic gene as well as secondary metabolites, and even for probiotic presence of probiotic related genes. For further, can you please do come poster number 23? Thank you.
My name is Imtiaz. I'm from INSTEM, from Dr. Arjun Guha Lab. In my research, I'm primarily focusing on understanding senescent cells. So senescent cells are very important in the etiology of many terminal lung disorders like IPF and fibrosis and COPD. And what we have observed is at 20 gray irradiation at 10 days, we have seen all the senescence markers are upregulated, which are shown here. Along with that, we have also seen TGF beta upregulation like ligand expression, TGF beta ligand expression, and phosphosmad along with targets like SMA and CTGF. What we try to understand, can we inhibit this TGF beta signaling, and what happens to the senescence phenotype? That was our question. We use TGF beta inhibitor HD208, and we allowed the cells to become senescent, and then we added HD208, and we saw that all the senescent phenotype has reversed. The cell size reduces, SAB-GAL and P21 arrest, and inflammatory cytokine has reduced. Next question we asked whether what happens. So why this? When we irradiate, what happens to this phenotype? How this phenotype initiated? So what we did is we chose different different type of radiation doses. We have seen that at 2.5 gray, the DNA damage recovers DNA damage, and this 20 gray we have seen senescence, and 80 gray is the apoptotic dose. And in 2.5 gray, you can see there is no DNA damage at 48 hours, but at 20 grays, the DNA damage persists, which is senescent phenotype, along with 80 grays, and that dose only we have seen phosphosmad. So that's why we are saying that DNA damage is essential for TGF beta signaling. And now, and in next, we are trying to understand what happens if we inhibit the ATM pathway, which is required for DNA damage response. Thank you.
Good afternoon, everyone. I'm Jayashree Ladke from Dr. Rashna Bhandari's lab, and today I'll be talking about polyphosphate in mitochondria. As the name suggests, polyphosphate is a long chain of phosphate residues linked by phosphoanhydride bond. It is a most conserved molecule, polymer after DNA and RNA, and it is present and it is well studied in bacteria and lower eukaryotes where the polyphosphate synthase and regulators are known. However, the polyphosphate synthase in mammal is still unknown even after 50 years of this field starting. So this is our project started where we looked at the interactome of inorganic polyphosphate in mammals, and we observed that the mitochondria, lysosome, nucleus, and granules were the organelles which are getting, the proteins of these organelles were getting enriched in this interactome. And when we looked at the interactome, the mitochondrial proteins were actually the FOF1 ATP synthase proteins. But the main function of it is to synthesize ATP. So, so can it synthesize polyphosphate as well? So for that, we isolated mitochondria from mouse liver tissue and incubated it with the different phosphate sources, maybe ATP or free phosphate. And we observed that overall polyphosphate synthesis were occurring in this mitochondria. So we suggest that, or we are asking whether FOF1 ATP synthase can also be a polyphosphate synthase. So we have further proved it that it can be the, for that you can visit my poster number 25. Other than that, next question which we asked was about the regulation of inorganic polyphosphate. And there is another phosphate-rich molecule our lab works on, which is IP7. It is synthesized by the protein called IP6 kinase. And we have observed that deletion of IP6K1 have shown the overall reduction in its respiration, suggesting that IP6K1 or IP7 regulates mitochondrial health. And what is the effect of this gene on polyphosphate synthesis of mitochondria? So for that, we isolated mitochondria from mouse liver of wild type and IP6K1 knockout mouse, and we observed that the polyphosphate synthesis activity of the mitochondria isolated from IP6K1 knockout mitochondria was reduced, suggesting that IP7 has role in regulation of polyphosphate synthesis in mitochondria. For further discussion, you can visit my poster 25. Thank you.
Now may I invite poster number from 26 to 30. Please come forward.
So I'm Kalyan from National Agri-Food Biotechnology Institute, Mohali. Our lab is interested in developing various sensors for food contaminants. And like in this work, I have developed a sensor for arsenic. So we have developed a material that is color changing material, and it shows a different color in the presence of arsenic. This is a bimetallic material, and it actually when the arsenic exposure is there, or in this sample there is arsenic, the structure, as you can see in the TEM images, the structure of the material, it gets degraded, and the photometric sites, they get exposed, and the color change is visible. So ultimately we have developed a strip based assay on which this material is being drop casted and dried, and once this strip is exposed to the arsenic contaminated food, water, or groundwater, like these things, the color change is visible, and we are able to detect as low as 0.04 ppb of arsenic, whereas as per WHO the permissible dose is 10 ppb. So other than this technology, we are also developing technologies where we are sensing gluten, because many people are showing symptoms of gluten sensitivity. We have developed some thermochromic materials where we can record the changes of temperature change on the temperature sensitive goods like food and storage during transportation and storage. Thank you. I'm open for more discussion.
A very good afternoon to all the dignitaries and the scholars present here. My name is Khairem Devi Kachenu. I'm working under the supervision of Nanaucha Sarma, IBSD. So my doctoral work is regarding plant Aziratina adenophora for its immense therapeutic potential as anti-cancer, anti-diabetic, and diabetic wound healing activities. So as you know, the Aziratina adenophora is a weed is very commonly available all over India. So why don't we make this weed into something very economically to put into economical use? So taking up this plant, I have studied anti-cancer, anti-diabetic, and diabetic wound healing activities, because these activities have been reported all over India. So just to validate it, I have to check it. So I found that indeed, actually, this plant is having immense therapeutic potential. So, yes. So, this plant Aziratina is in my study has potential in managing CRC, that is colorectal cancer cell line, and T2DM, that is diabetes mellitus, and diabetic wound healing. So this study underscores the importance of exploring natural remedies for complex health conditions, paving the way for future research and potential clinical applications. And it has been found that T2DM and this cancer coexist, and the prevalence of this is very common globally. Therefore, the research in understanding and defining the biological relationship, as well as the molecular complexity between these two medical conditions, are necessary. For more details, please visit poster number 28. Thank you.
Good afternoon, everyone. I'm Khadashri from IBSC Imphal, and I'm working under the supervision of Dr. Sudipta Das. And my work is on characterization of capsicum for value capsicum from Northeast India for value addition. As we know that capsicum, that is chili, have a high economic value in India, and in the Indian economy, it provide around 540 billion Indian rupees in the economy, and more than 3 million farmers are livelihood depends on the cultivation of chili. So moving on to the Northeast India. Northeast India belongs to the biodiversity hotspot, that is Indo-Myanmar biodiversity, and it is home to the many endemic species of chili. And one of the most chili is the Capsicum chinensis, that is the Butchalokia, 7 hottest chili. But our work focused on the aromatic value of chili beyond Capsicum chinensis, that is one of species of Capsicum annum, which have a very unique aroma, that have a sweet and smoky aroma. So our work focused on the biochemical profiling of that species, and studying the expression profile of the aromatic pathway and the carotenoid pathway. So for more detail, please visit poster number 29 on the first floor. Thank you.
Good afternoon to all. I am Khushbu Kaushal from THSTI. So my research work focuses on the role of maternal inflammation during pregnancy in associations to fetal growth restriction. So FGR is a condition where the fetus did not grow properly to its biological potential, and it is the FGR babies are at a higher risk of developing neonatal mortality and under 5 mortality, and also metabolic complications later in life. So for studying maternal inflammation, we have used cytokines as a surrogate marker for the inflammatory responses, and we have quantified 24 markers at a different time points longitudinally in a pregnancy and compare them between normal and FGR pregnancy. We do identify that the placental growth factor at an individual level was the only cytokine that was found to be different in FGR. Interestingly, we found that there were some of the cytokine clusters that was specific to either normal pregnancy or FGR. So that can be used as a predictive marker and can be validated further to that can be used as a predictive marker for FGR pregnancy and to predict at a other high risk pregnancy. So other than that, we have also generated a cytokine profile longitudinally in a healthy pregnancy that adhere to a reference value for Indian populations. For more information, visit poster 30. Thank you.
Now may I invite poster number 33 to 40. Please come forward.
Good afternoon, everyone. My name is Vimi, and I'm from IBSC Imphal, and I'm working under the supervision of Dr. Nanaucha Sharma. And my work, the title of my thesis is Paris polyphylla, which is a dynamic herb for treating diabetes and its complications. So we all know that diabetes has various complications associated with it. Traditionally, we know diabetes is associated with various retinopathy, kidney diseases. But however, recent studies have shown that new complications are arising from diabetes, which include cancer and pancreatic cancer, liver cancers, and various types of cancer. So this plant is a very interesting plant, which is found in the northeastern part of India. While it is very popular in China, in India, it is exploited for trading purposes only. So we are focused on using this plant for exploring its therapeutic potential. And we have found that this plant has not been used for treating diabetes. And we have found from the traditional knowledge that this plant is used for treating diabetes. So we explored how this plant can be used for treating diabetes and its complications like colorectal cancer, and also for treating diabetic wound healing. And we have found that this single plant can be used for treating colorectal cancer, and also it has anti-diabetic property. And we have prepared an ointment using this extract, and we found that it has diabetic wound healing activity, and it accelerates the diabetic wound healing activity in just 18 days as compared to standard. For more details, please you can visit poster number 33. Thank you.
Good afternoon, everyone. Myself, Rajita, working under Dr. Anand Srivastava from NIB. So my research topic entitled Insights into the Molecular Events During Feeding Stages in Ticks. So these ticks are tiny hematophagous ectoparasites. They stay outside the host body and take the blood meal. So in the process of taking the blood meal, they transmit various pathogens. Those causes the various infectious diseases. So these diseases, because of this livestock industry is getting huge economic losses. So how to avoid? So main tick research, what is the purpose? So that purpose is to how to avoid the tick infestation. So to understand this, we need to know more about the what is the tick and host interaction, what is happening in this interface. So for this, we have used a transcriptome, proteome of salivary gland, midgut, and ovary. These three tissues are the main organs of the tick. So we use this multi-omic approach, and we annotated many antigens which are like hidden in the salivary gland extract, those which will go into the host body and suppress the host immune response. Thereby, it will help in the continuous feeding of the tick. So we many like proteins we have identified and we annotated its function. On the other hand, like a complement is the first line of defense against any infectious pathogens. So when tick infest, it is suppressing the host complement system. But what are those molecules which is infecting this host complement is not known in Hylomonitolicum tick. So we have evaluated its anti-complement activity and we confirmed as this salivary gland extract has anti-complement activity, and we used this multi-omic approach like transcriptome data of Hylomonitolicum tick, and using the mass spectrometry analysis we identified one of that antigen which is inhibiting the host complement system, and we named it as a hampin-1. So how from omic to how we achieve like one single protein, please do visit poster number 34. Thank you.
Good afternoon, everyone. So my name is Mehek and I'm from National Center for Cell Science, Pune. So my graduate work involves understanding the role of clathrin light chains in early mammalian development, basically by regulating actin cytoskeleton and cell signaling. So to begin with, as you all know, the early mammalian development starts with the formation of a zygote, which undergoes several mitotic divisions and form a structure called blastocyst. So as you can see in this above diagram, so blastocyst, the inner group of cells called ICM, which ultimately leads to the development of the whole organism, and outer layer is called trophoectoderm, which forms the placenta. So this early development is important to study because to understand the underlying mechanism of cell fate specifications. So it involves various pathways, cell signaling pathways, one such and other cellular and biological processes such as endocytosis. We particularly work on clathrin-mediated endocytosis, where the major player is clathrin, and it has two components. One is clathrin heavy chain and other is clathrin light chain. My project particularly focuses on clathrin light chains. It has two light chain. In higher mammals, we have two light chain genes. One is clathrin light chain A and other is clathrin light chain B. And it has a binding site for actin in a cell through which it can mediate actin organization. So as I mentioned, that I work on clathrin-mediated endocytosis or intracellular trafficking. So it is involved in regulating cell signaling pathways as well. So first question, I would like to address how these light chains can mediate cell signaling, as well as how they can regulate actin organization in cells. To address the question, we made clathrin light chain knockout lines in mouse embryonic stem cells using CRISPR-Cas9 genome editing system. And the key findings from my project is basically, if you can see wild type cell, you have proper actin organization as well as proper Wnt secretion. But when you remove clathrin light chain A or both the light chains, you can see altered actin organization as well as reduced Wnt secretion. And you can activate Wnt signaling either by providing the ligand or restore the actin organization either by overexpressing the CLCA. So for more details, please visit my poster, which is poster number 35. Thank you.
Hello, everyone. Myself, Neeraj Vasnik from Regional Center for Biotechnology, pursuing my PhD under Professor Shivram Mailavapu. So we in our lab are trying to understand the basics of cell division governed by molecular motors called dynein. Using proteomics approaches, we have found a potential candidate which comes as a reproducible interactor in the cell division, which is this protein called T2. So T2 is an actin bundling protein. It is involved in the actin mechanism in the cells. Using knockdown approaches, we have validated its functional significance in the cells. Using high resolution confocal microscopy, we have seen its live localization in the cell division dividing cells. And also we have used mass spec-based proteomics to understand the various protein-protein interaction it makes in the different stages of the cell division. So the crux of my findings are that translin 2 is this T2 protein or the translin 2 protein is involved in maintaining the metaphase plate of the dividing cell. Not only that, is also involved in the chromosome congregation where the cell fails to form the metaphase plate. Metaphase to anaphase transition, which is the checkpoint silencing, this protein has also shown its role in there. Not only that, centrosome, which is a membraneless structure, This protein has also showing upon knockdown condition that the integrity of the centrosome has been compromised. Last stage of the cell division, which is the cytokinesis, where the using the abscission stage, the cells separate physically from each other. This protein has also shown its involvement in these cytokinetic stages as well. Another thing is that being an actin binding protein, this protein has also shown its interaction with the other counterpart of the cytoskeleton, which is the tubulin. So to know more about this mechanistic details, please visit the poster number 36. Thank you so much.
A very good afternoon to all. My name is Nikhil Hanumantrao Samat. I'm from National Center for Cell Science, working under the guidance of Dr. Shailaja Singh. So my project overall revolves around the EGFR-mediated autophagy signaling. As we have seen that the most of the cancer therapeutics are going resistance because of the EGFR mutations and eventually inducing the autophagy mechanism. And nowadays, it can be said that autophagy is emerging as a newer cancer hallmark. So based on that, we make use of systems biology, exome sequencing, or say the NGS sequencing data to build a reconstructed mathematical model which defines the EGFR-mediated autophagy signaling. And based on the analysis of that mathematical model, we can get the crosstalk points. And by modulating those crosstalk points, we can say that if we modulate those crosstalk points, the whole system might collapse. So based on that, we read some articles and found that there are some plant organic compounds which have been used to treat the mutated EGFR or the resistant EGFR therapeutics. So we found certain anthraquinone derivatives which have been used for treating those patients. So we make use of machine learning to scan or dock almost more than 12,000 residues or small molecules to search for a single residue, which was the A52, which we considered as our investigational drug. And further, we have done a bioinformatics analysis, the docking, molecular dynamic simulations to check the efficacy of that. And also we have done certain in vitro and in vivo analysis to check whether the therapeutics or the A52, which we considered as our investigational drug, is working efficaciously or not. And the results are quite satisfactory that yes, it is working enough to block the tumor development. In the in vivo, we can see that. And also in the in vitro analysis, we have done the ICC, the Western blot, and the apoptosis assays and the cell cycle. We can see that yes, it is able to block the growth of tumor cells. And we have also tried to combine it with the standard available drug, which is the EGFR TKIs, and the standard autophagy inhibitor, which is chloroquine. And I have more things to explain. Please do visit my poster number 37. Thank you.
Good afternoon, everyone. I am Piyush here from ICGB. So my work involved redesigning E. coli strains for bioproduction. As we move into further into 21st century, we face the biggest challenge of climate change, and we need to redesign microbes as they can help us to produce from products like antibiotics and food, even to meet the growing demands of our population. So we went on to analyze various strains of E. coli that are available for bioproduction, and we found that they use the various carbon sources which are available for cheap, like glucose and glycerol, and we found that there is a huge difference in the glucose uptake rate and the production rates of various compounds like succinic acids and ethanol. So the next question was why it is happening and why they are why the strains have a difference in their various various uptake rates and production rates. So we use 13C MFA, which helps us to dissect the metabolism of these strains. So as you can see that there's a difference in the central carbon metabolism of these strains, and various pathways like EMP pathway and pentose phosphate pathways are differentially being active in these strains. So the next step was to find out how we can change these pathways. So we went on to do LCMS analysis of these strains to find out which proteins are being regulating, which are regulating these pathways. Do they have any role to play in the activity of these pathways? And yes, we found significant differences in the central carbon metabolism pathway of these strains and the regulators of these pathways. And next step was how we can use all this information in redesigning these strains. So we turn our attention for constraint-based modeling, and we tried to integrate these LCMS, these proteomics data and metabolic flux data into constraint-based models, and see if these models can predict us some good targets for genetic engineering of for enhancing any products like succinic acid or ethanol. So for more information, you can visit my poster number 39. Thank you.
Good afternoon, everyone. I am Pooja from RGCB. I'll be sharing few insights from our work on deciphering the role of RFX1 in regulation of cancer stemness. RFX1 or regulatory factor X1 is a transcription factor that promotes differentiation and is downregulated in most of the cancer. So our hypothesis is that the presence of RFX1 in cancer stemness cancer stem cells may allow them to differentiate to cancer cells that can be targeted using chemotherapy. And we have used Entera cells for our study. Entera cells are cancer stem-like cells that can be differentiated using RA treatment. And the differentiated Entera cells showed an increased expression of RFX1. And further to study the effect, RFX1 was overexpressed in Entera cells. And after stable cell line generation, we could find that RFX1 overexpressed cells showed reduction in proliferation, viability, and migration and invasion properties. Moreover, self-renewal and stemness properties were also compromised in RFX1 overexpressed cells, which was studied using flow cytometry of cell surface markers. In addition to this, we could find that pluripotency marker OCT4 was binding to RFX1 promoter and downregulating its expression. And OCT4 immunoprecipitation followed by mass spec analysis showed that HTAC1 was associated along with OCT4 in mediating RFX1 downregulation. More interestingly, we could find that RFX1 downregulates LRH1 and Frizzle5 genes that can promote OCT4 in turn. So there is an interregulatory mechanism between RFX1 and OCT4 in Entera cells. And we could find that the treatment of Entera cells with romidepsin, which is an HTAC1 inhibitor, could increase RFX1 expression, thus indicating that the disruption of regulatory mechanisms have therapeutic potential. Please visit poster number 40 for more details. Thank you.
With this, we finished 40 flash talks, and there'll be posters corresponding to these numbers downstairs, ground floor, first floor, and second floor. And now we're moving to the next exciting session, which is a panel discussion for which I would now request the distinguished gathering here, Dr. Rajesh Gokhale, Professor B.J. Rao, Dr. Rachna Tripathi, Dr. Krishna Ella, Dr. Apurva Sarin, Dr. Vishwanathan.
While we are waiting for the chairs to be assembled for the distinguished invitees to come here, this is your one chance for all the students to give back the GAN or ask very tough questions to the people on this side of the podium. Because I think we have given enough big GAN to all of you. Any important burning questions that you have, be it science or philosophy, you free to ask these questions, and I hope our distinguished guests will be able to answer them. Please engage. And in case the students don't engage, I will request the panelists to probably trigger those conversations so that it becomes more dialogue-based thing. So please, students, I request you to kindly participate as actively as you can. Thank you.
Now I'll request this session will be moderated by Dr. Murli Basham, Dean Cdfd. Murli.
I kindly request the panelists to please come on to the dais. Thank you. So as our director.
So before Murli takes on, I also request the directors at the end of this session to also say, of course, your time will be limited to 30 seconds. because you can talk to your students at length in your own respective institutions, but it's also an opportunity for all of us to convey a particular information and message that we're all supportive to the students, and we would like to make sure that we are very keenly desirous of ensuring that our students succeed in the future. Thank you.
Okay, so the main focus of this student mapping conclave or placement conclave are the students themselves, of course, and we have 80 students representing all 16 IBRIC plus institutions. So these students have their own doubts, as our director alluded to. So what are the opportunities after PhD? Is it the traditional opportunity? Do you want to be an entrepreneur, or you want to go into some other field? You want to go to industry? So we have the distinguished panelists here who will be able to clear your doubts. I'll just briefly introduce two of them who were not introduced in the morning. Dr. Apurva Sareen, she is the CEO of one of the most prestigious granting agencies in India, the DBT Wellcome Trust India Alliance. And Dr. Vishwanathan, who's the Vice President of Biomanufacturing at IKP Knowledge Park. And we as Hyderabadis have a lot of prestige in knowing that we have IKP Knowledge Park in our city, one of its kind. The other panelists, of course, so please keep in mind, we have panelists who have been earlier or currently involved in policy making and are affiliated to various government agencies, including Ministry of Science and Technology, Department of Biotechnology, Department of Atomic Energy, and also the HRD Ministry. And plus, of course, we have the industrial stalwarts, Dr. Krishna Ella from Bharat Biotech, and also Dr. Vishwanathan. So I won't stand in between my young friends and my younger friends. So I request the students to start the session, please. So if you have any doubts or clarifications, this is the time. Yes, please. Please introduce yourself and then go ahead.
Thank you for the opportunity. My name is Shruti. I'm doing PhD from NIPGR with Dr. Jyoti Lakshmi. So I have a question to all of you over here, which is considering now that we are post-pandemic, where there was a boom in virology research. Me being a plant scientist, I would like to ask you, where do you think, now we all know where the research in India, where the topmost research or the most people, what are they doing research in? But as a plant biologist now, I would like to pose a question, where is the most funding going? And this is with the academic picture and also with the industries, private companies, in which direction the most funding is going? Where does the country want to, the government of India want us as researchers head to?
Maybe Secretary DBT should address this. Government funding.
Thank you for putting up this important question. I think there's a huge gap in between what government wants to do and what I think you need to understand the science and technology develops over much longer period of time than the investment cycle as well, actually. And that's where the biggest challenge is to synchronize the investment that government makes through funding and the science and technology outputs that come at a period of time, actually. So how to align these systems is the most tricky part. So for example, if you really look at, if you ask me what would happen to the food industry in say another 10 years of time, it's easy for me to address. If you say what will happen in the next 5 years, maybe not very easy for me to address, but in 10 years time, I can almost guarantee you that at least 1/10 of the food will start to come from the laboratories, actually. Because primarily the vagaries of science, climate change, and these kind of activities will continue to grow. The challenges will completely be in terms of productivity as well as there are various components involved in agriculture. Agriculture is one of the most complex sector because the labor is involved in it, the mechanization is involved in it, variety of other players, stakeholders works in the system. And all of these will undergo a transition in life as the technologies and science will continue to grow, actually. So if you want to tap something in 10 years time, I would suggest, you think of ways in which useful molecules that are produced by plants, which are actually harnessed for variety of other purpose than food, will in fact become a reality from the laboratory setting in terms of manufacturing, because the land for the food and other aspect will actually become more and more as the population continues to grow. If you want to look at an agriculture from the food sector point of view, actually, you already must have heard in this political rallies, now Annadata, ab aap, pehle aap Annadata the, ab Urja Data bhi ho gaye hai. Correct. So what does that mean? That you're going to start agriculture crops for energy. So biomass to bioenergy will become also a reality, because that's the way transitions will occur. But again, these will happen over 5 years period of time. So it depends on who you are. I mean, if you are a PI looking for a 3-year life cycle, you will look at, what I would like to do such that I can achieve in 3 years time. If you are a company, you are looking at making finances, you will look at what will you do. So there are various strategic areas, sectors actually will happen. And I think, in my opinion, the agri-food industry will have maximum amount of transition in next 25 years, because the predictability of food and agriculture products to come from will have to be mixed from both lab and land. And I think that's where the transition will occur. But immediately, if you ask me for an immediate, what can we do actually? Any program, for example, genome edited program lines or whatever areas you look at actually, obviously you should look at the best way to start a career right now is to look at what are we importing the maximum number of times in looking at actually. For example, the challenge is, can we take up this within 3 years period of time? So for example, oil seeds, oil of every part of the oil industry, I think more than 10,000 crores of oil is being imported right now. Whereas we were the surplus industry, whether it's a groundnut oil or a mustard oil or any of these oil. So many of these sectors will continue to, very rapidly you want to make an impact. And this is a sector to grow actually. But that's why I said, the funding is not that easy to say and what you want to achieve. There are these kind of mismatch in terms of where the science develops, where the technology develops, and where the funding emerges actually. I hope I've addressed some of these issues.
Hello, everyone. I am Binduma Yadav from Nabi Mohali. Panelists, when we are seeing a doctoral graduate, what are the skill sets that you are looking for?
So you're addressing government fund like opportunities or private or both? Both. Okay. So let's start with
I think
Professor Rao.
it's a very, very ground level question and a very relevant question. What type of skill sets you have to have? I think there are many, many answers for this. I think one of the most important answers is to be able to predict some of the big things that are going to happen in biology in future. You are at one point of time where you are acquiring skills, but your training should be so broad-based that you should be able to predict 5, 10, 20 years from now where the field is likely to go. That's a very important requirement for all of you so that you can strategize. Based on that kind of prediction, it is possible to, sort of envisage what sort of skills you should have to map yourself there. So it is entirely up to how you think about it. For example, the current pharmaceutical industry essentially depends on fossil fuels. Most of the chemicals come from fossil fuels, which has a very high carbon footprint on us. Most of this has to move away from fossil fuel to biofactories, microbes. And all these intermediates, important intermediates, must be able to synthesized in the biofactories. For that, we need a lot of engineering skills. We need to be able to tinker many things. We need to be able to compute many things. We need to be able to think about many scale-up operations. So this is just one example I'm giving you. There are many other examples. So your ability to predict the future that biology unfolds and how you are able to map yourself, I think is a good exercise for all of you to go through. You may succeed, you may not succeed, but certainly it's an interesting exercise to go through. This is all I can tell at this stage.
Dr. Ella, you want to add something?
Skill set is your personal choice. Your personal choice. Nobody can enforce, you have to become good. Okay? Bottom line is that what makes you passionate of doing anything. For example, I don't want to give your interest, but I'll tell some of the poster presentation. Somebody give a tuberculosis, multi-drug resistant, all the markers. One can look at it on the entire spectrum of what are tuberculosis resistant strains in the entire map in India. How that can be applicable in a clinical research when you're doing in a tuberculosis vaccine trial or a multi-drug, antibiotics or whatever you're trying. How that one skill set can be applied in a bigger, broader aspect. The kid has to visualize that, and nobody can tell you what you should do. It is your choice, but how that skill set can be applied to that. So,
Dr. Apurva, please.
Thank you. I just want to add to this to say that you've heard a lot about what the expectations are on your group. It's also important to just reiterate what the previous speaker said. You must find your own place in that larger, options that are being offered you. Your training may have been in a particular model system or an experimental system, but it has really more importantly trained you to deal with the uncertainties and the challenges that experimental system provides you. So perhaps today it is important to be able to pivot, to use your knowledge and your training into areas that you think are perhaps very exciting, because it is not ever too late to pick up a new area. I mean, some of the most prestigious fellowships that support young people are fellowships that support transitions into other disciplines. And that is for good reason. It is really so that you can keep skilling yourself as you go along. So I don't think you should see this as some place where, it's a do or die decision. It's a milestone in your lives. And I think everyone in the room who's anywhere near submitting or has submitted has to be congratulated for coming through probably one of the most eventful periods of your life. So don't be too frightened, and ask the questions that allow you to make decisions. Don't worry too much about, like you were saying, the most
Mike.
Okay, sorry. So I'm audible now.
Keep it closer.
Okay, tested. Yeah. So now we know that climate change is the most serious issue we are facing. So we have National Action Plan on Climate Change in 2008 in India. So now we are in 2024. It's almost like 16 years. So as a DBT institute, like we are all from DBT institute. So what initiatives there are in DBT institutes just to mitigate such scenarios? Like we talk about IT boom in 90s. That was because of LBG reforms. So what kind of reforms which we have now to bring that boom in our field, in scientific field? First question I want to ask that. And second question is, like you said, 4% of GDP is contributed by research. That means we have cost to that output ratio is 4. That means 16% of funds must have gone to the research. So that means we have bio-waste issue also. So why we don't talk about the recycling research? We always talk about recycling economy, recycling that and this. Why we don't talk about recycling research and sustainable research? I would say that would be a great initiative, because it's a 4% of GDP. That means we can contribute significantly to mitigate climate change. So what policies we have in place to address such issues?
Well, thank you very much for asking that question. In fact, from DBTs for last almost a year or more than a year and a half, I think we've been taking up various meetings across all the institutions in the country. In fact, we had close to 16 meetings related to this, of how biotechnology can solve the problem. And obviously, there are no easy solutions. I mean, the real and the biggest problem in all these is capital. Who's going to finance transitions? I mean, that is why, I give an example. For example, if you look at milk industry, just think about milk. The domestication of plants and animals was the basis by which the life was started. If I tell you that the milk industry in 20 years will transit to a lab, do you think you'll agree with it today? No, because it's already happening. It's already happening, but it'll completely, maybe almost 100% may move out in 20 years' time. Milk is a formulation, right? If you really look at from a biochemical point of view, it's a formulation of couple of proteins and other ingredients, and those are the kind of putting together actually. So how are these transitions and why this is important? Because the greenhouse gases that are produced from the veterinary animals actually, and the amount of time and the amount of energy that is really required to produce the amount of milk that is required in the country is enormous. But the cost of that milk is very high. The cost that you produce today is not what cost that you would like to buy actually. If I ask you buy 1 liter of milk produced in the lab with low greenhouse gas impact, you're not going to be paying for it actually. So how do you do these transitions? And every country in the world is actually debating about this. There's a very nice review article in a recent Science in the Nature actually from a Netherlands group, which is Netherlands is a very, very forward-looking country you should look at. It talks about questions, it talks about ideas, it also talks about this activity. However, what India is doing, maybe you may hear it this next week, you may hear something from the PMO's office actually, and hopefully, it will have some impact. I don't want to say it loud now because, it's kind of under the process of cabinet approval. But I think there are distinct, for example, you look at our Prime Minister's life, which was the whole idea is what actually? Life for lifestyle for environment is really based on the circular economy model actually, is the reuse the things, don't waste things actually. So if to contribute simple thing, but R&D, putting money into R&D is very, very complex challenge actually, because the financial aspects are not very easy to look at. For example, I'll give you an example of 2G ethanol actually. I don't know if you follow this, the model. I mean, government of India is talking about 20% blending. What is the cost of per liter of ethanol that government is costing right now? Do you know? 2G ethanol? What does it cost per liter? It's about 140 rupees per liter. That's what is going to cost. What is in the markets being sold today? It's 60 rupees per liter. So the government is giving a subsidy to this account for this technology to develop. So at every sector, one will have to come up with such policy changes and implementation to make this wider impact. It's not very easy. It's very important, but it's not very easy to do it. In fact, this paper that I referred to, you should go back and look at. It talks about if I want to shut down petroleum industry, how much will it cost? And you look at the amount, gigantic amount that it talks about, and then you'll realize that why this is such a complex problem. But you are absolutely right. I mean, we need to understand how to go through this transitions, I think.
Saifi, sorry, I think we'll go to the next question. We can come back to you if we have. Okay, yeah, go ahead, Saifi.
Communication departments in all DBT institutes nowadays. Why don't we set up a climate mitigation department? Let's just talk about even today. We might have printed so many posters. That is a plastic. We should go for alternative. Like we can put a screens where we can put a pen drive and our poster will be displayed. It will be a one-time investment, but we can reduce the plastic use, which we do daily. If you go a printer, daily so many posters are printed. They are just one-time use. So that kind of initiative, I think we should take at least just to reduce the plastic.
The first program that BRIC announced was zero waste campuses on the DBT institutions actually, and that was the first program that was announced on the day when the BRIC was formed. The whole idea was exactly that. Can we implement in practice is obviously one has to look at. But I think that's what I think. For example, our institution names also we are now changing. NABI is actually what if you want to talk about?
Hello, this is Ashwini from NABI. The mandate of the whole institution is changing now. There are two institutions, National Agri-Food Biotechnology Institute, which primarily focuses on primary agriculture, food security, nutrition, and there is other institution which talks about secondary agriculture. We thought of combining the two things and come up with a new idea that we can do biomanufacturing in the institution related to agri-food. So the name of the new institution will be National Agri-Food Biotechnology and Biomanufacturing Institute. And with the help of Department of Biotechnology and other support, we are trying to develop all these things we are talking about, how to convert the bio waste, which is being generated in farms and beyond, into some useful products. So I think a lot of efforts are going on at all the institutions. And in case any of the student would like to talk to more about it, I'm available for the whole day. Thank you.
So Dr. Gokhale, this is the NCR new institute you showed in your, no, this is Mohali. Oh yeah, okay. Yeah, please. We had a question there.
Hello, my name is Shabnam. I'm from National Institute of Immunology. My question is to Dr. Gokhale, DBT Secretary. So BioVision is a great initiative, but that vision needs to be like communicated to the other part of the society, which is not scientifically literate, we can say, so that the transition that we are talking about can be easier. So do you have any plans to include biocommunication per se in your BioVision, so that any like transition from lab to the life that might change their that might change any that might change the society as a whole?
It's very important. Maybe I'll let Dr. Krishna address that. I mean, communication is a very important aspect of life. I mean, the reason why I'm asking Dr. Krishna or her to talk about it, because, when you started companies, I mean, both of them initiated companies at a time when biotech sector was not at all in the horizon, actually. And I think how they spelled out to government that, provide us some kind of benefit, or whether they did provide, I don't know. Maybe I'll ask Dr. Krishna and then you to talk about it, and then I can address it.
Let me tell you honestly, I'm now sustaining only because of the government funding. Let me tell you honestly. Okay. Technology Development Board funded the initial startup. Okay. That's how I made an entrepreneur's journey, because no private equity want to invest in a private, because they don't believe in entrepreneurs in this country. No private equity. Okay. They only believe on as a loan, not equity guys. Leave apart that. But I think, the scientific communication is happening in this country. Nothing to worry about it. If you're not scientific communication had happened, you would not be a PhD student today. Okay, am I right?
Can I add something to it?
Sure.
So once I met someone and he asked me, that guy asked me like, what are you doing? So I told him like briefly what I'm doing. So when I told him, he was like, oh, this is not this, this is that. So scientific communication is happening, but I think we are restricted till the journals, but not to the society as a whole.
Same question is arising even in US. Even people doesn't know, they only know New York Times and Washington Post. Even there, they don't know anything. Trust me, I'm telling you. It is not the Indian phenomena, it's a global phenomena. Okay. I mean, what is important is just the scientific impact that has happened. See, we are also not able to communicate. Like for example, in vaccine field, we saved maybe $5-7 billion of import of vaccine into this country from other companies selling it, okay, by having domestically done. And now we are able to export the vaccines to 120 countries, okay? So there is an impact, but the impact is felt this thing, but the communication is very tough. And only the Times of India and all these people has to communicate, but they communicate wrong thing about India all the time. Okay, anything they make it negative only, they get news attention in this country, not the positive news. You tell biotechnology we've done something like Gokhale said, done something great, a small piece will come. But Gokhale said India is not doing this job, he'll say, immediately it becomes sensationalist in the first page. We have also scientific communication with the journalist, has to be educated. I think you should go and educate the journalist first. Thank you.
Dr. Rachna.
I think he has given a good answer. At present, I don't have anything extra to add.
Yeah, and I would highlight one of the student talks, Devon, she's from Cdfd. She had actually talked about science communication. Maybe you can visit her poster today. Okay, next question. Okay, yeah, please.
Good afternoon, panelists. So my name is Brahmamay Basu from RCB Faridabad. So I want to ask a very baseline question. So there is a myth that after PhD, you definitely need to do a foreign postdoc to make it big, make anything to happen. So today I was actually felt good when ma'am told that she didn't go for a postdoc and she is doing such a great thing. So I just want your opinion on our pathway. Like now we are thinking of what to do next. So is it mandatory to do a foreign postdoc to actually make it big, or it's like, no, you can't do anything? It's like very baseline question, but I want your opinion.
Yeah, Dr. Rachna, and then I would request Professor Rao also to add, please.
That's what I would like to begin with, that whether if it's a academic career you want to choose, Professor Rao should answer it. And but for the private career, or for me, we give really good training during the PhD. The exposure is good, and there has to be some time where we stop being student and start working, because when you work you learn more, you have more idea that what kind of field is growing and how can you apply your knowledge. So that has to start as quickly as possible.
One general red flag is the following. If you start following what your surroundings want you from, there is no end to it. You must ask a question yourself, what is that I want to do after PhD? The answer must come from within, and that will be the best answer. So your answer, you should be able to discover, and then options open up. If you go and ask X, Y, Z, you will get A, B, C type of answers. It will confuse you, disorient you. So the best general answer for all of you is to start asking questions now that you are finishing your PhD, what is that you want to do? What is your passion? Where is it leading you? It's a very relevant question to ask now.
Yeah, Dr. Vishwanathan, biotech industry perspective.
Since you mentioned about BIG, I have actually an example from BIG. BIG stands for Biotechnology Ignition Grant. We have around 20 students after PhD, they have received this funding program. This is a 50 lakh grant. And since somebody is talking about sustainability, I will take one example. Student immediately after, this is actually even before PhD. So they received 50 lakh grant to convert agriculture waste into activated charcoal. And while doing this program, they received a 10 crore Prince William Earthshot Prize, and they received 39 crores funding from Elon Musk Foundation. So it's not about, if you want to make big, whether to postdoc, what you want to achieve is more important. I have several such students, after PhD and after postdoc from University of Hyderabad. We have a company, BICUS. It stands for Bi Central University Scholars. So these 3 postdocs came together. Now they have raised around 50 crore funding, and they have 60 people. They went to Andhra University, recruited entire batch of students to bring. So they can create a huge impact with these 50 lakhs or 1 crore or several grants. I'm just I work for BAZ grant in BIRAC and DBT associate programs. So I'm just referring, but there are several programs. BIRAC BAZ is a flagship program. There are 1,000 students who received, and just from IKP there are 193 students who received this funding, and they raised 930 crores. And in the afternoon, I'm just talking about 1 or 2 examples. Maybe I will talk more, and you can discuss about this.
Thank you. Yeah, please, Dr. YouTube.
Outside?
Yes.
I didn't know that. I thought only very quick. You are a fresh graduate, you have some skill set. I have a 3 years postdoc from, say, from Stanford, okay, whom I'll hire. I will hire you, not him. You know why? That guy will come with the US by thinking that I'm a brilliant scientist. I got in Stanford with ego and all that. And you are a young fellow and is already 3 years old, actually. Okay? So I will prefer you than the world. Thank you.
Okay. Yeah, I timed it at 25, but maybe we can take 5 minutes more. We have 5 more minutes. Yes.
Hello,
Thank
sir.
you.
Myself, Sadanam Bhanuse from NCCS. Sir, Dr. APJ Abdul Kalam ji said that invention, innovation, and implication make the country at bigger height. So we give the training to the PhD student here, and intellectual and good people, researcher, go to the abroad for find the 2 answer of the 2 questions. One is that we can get the economical stability by getting the more dollar, and another one is the exposure of the new knowledge, new technology. How the BIRAC and DBT can explore that economical condition here and knowledge and new technology to stay the all intellectual people in India and make country to the bigger height.
I thought, I tried to talk about the same thing during my, I think, let me just give you a brief example. I think it's a very important question to understand, as which I was trying to talk about, why institutionalizing activities does make a big impact. You'll be surprised, Department of Biotechnology is a very unique to very few countries in fact, have a focused department in government for biotech sector. Very few. Now, because of this and because of various activities that happened, for example, India Alliance that was set up in 2008, in 2014, actually, then BIRAC was set up, and now BRIC is set up. How various institutionalizing processes now have enhanced the capacity and capability for us to do things. That we have now reached a stage where we're starting to talk about, can we set up a mandate for the world to lead? And I think that's where the transformative confidence that are starting to emerge among our people and our ideas. And I think that's where DBT has been putting as great efforts and emphasis. And, for example, as he said, for the big grant, actually, I mean, this is one of the most flagship grant. I mean, I think we have a kind of a impact of 8,000 startup companies where we are connected with right now. Now, the challenge is to make them grow and make them reach a stage where the revenue starts in their companies. Now, how to be able to do this requires one more level of activity, which is what we are trying to do through biomanufacturing. Actually, you bridge the gap, you kind of handhold one more level of activity, and eventually then you become a commercial activity. So there are several activities that are going on with department actually. And I think what you said is very, very good, but it's a bigger challenge to do. But I think it's been happening in the country actually.
Yeah. Hold it closer and shout. Okay, I'll shout. No, hold it closer and then you shout. Yeah. It's not on, I think.
Yeah, sir, my name is Jagadish Ganla. I'm representing Federation of Asian Biotech Association, FABA. So I want to ask one question on behalf of all PhD students. So my PhD is from Germany, and I compare that PhD with Indian PhD. When I talk to many of the PhD students, the first thing that I see is depression in their face. What they complain about publications and 7 years, 8 years, 9 years PhDs. One thing that I want to ask all the panel here is that how can their PhDs be happy PhDs and how can they be creative when focusing on, especially when we talk about biomanufacturing, all the big things we talk, but when you really talk about, look, oh. I mean, this is a bigger concern, right?
I think the I3C program that I talked extensively about is about students. It's looking into the careers of students, making the careers of students, giving them a path about it. If you get absolved yourself, and this is typical of all PhD students in India, and very much typical of Indian classical syndrome, which I call, is that when in doubt or when in worry, you go inward. Right? You start thinking that I am the problem. I am going to solve the problem. I don't need somebody help. When you are happy, you share. When you are sad, you in fact go inward. In the US, the model is completely different. You're very happy to talk to people and convey to them that, no, this is not happening, whereas we are ashamed of saying something like this actually. And I think this whole challenge is because we are not working in a bigger networks. We're working in a smaller labs. And I think this I3C program, in fact, I really believe that I think it's a transformative for the 19th century PhD program, did its great service to the country by creating human resource, but now it's time to solve problems and solve real world problems actually. And that will motivate everybody. And the other point that you talk about is very much of an individualistic actually activities, which is to do with the fact is that the way we treat our PhD students is like a student. And that's why, the first statement of mine, treat it as your first job. You start to become colleagues as it's in the Western labs, actually. This will create much happier people, much happier life, and, more happy faces, as you rightly said, actually. Thank you.
Thank you for that question. So I think we'll close, but I just wanted to.
I just want to add here one more thing to this. I thought it's very it might be very useful for the students, that we should also start like some industry and academic collaboration where the local industries will have some small problems which they are not able to solve, or they need your help, or academics can spend more time on it. If some such small problems are taken from different industries to the labs, and students are, even if they have a major project, along with it they are working on such problems, probably they understand the ecosystem better, as well as they are prepared well for the outside world. That's my suggestion.
Okay, thank you. So this question actually reminded me of something which I was supposed to say in the beginning, but I forgot. So there is published research which shows that if you have a PhD from India, you have a better chance of landing a job in India and abroad, and also to have a better package compared to a master's degree. So that is published research from India. So with that note, I thank all the panelists. Please give them a round of applause. And I also thank all the students for engaging in such active discussion, and a bigger round of applause to the students, please. Thank you. I now request Dr. Yatish Achar to present a memento to Dr. Apurva Sarin, please. Yeah. Thank you. So I request our director, Dr. Ullas Kultu Sitaram, to please moderate the next session for the director's presentations.
So there's no presentation. Please don't worry. I know we're all hungry. If the directors want to speak 30 seconds, please be crisp. If you want to add anything, please do it. Otherwise, yeah, anyone. Yeah, Shubhra.
So first of all, I think it's a great opportunity which we never had in when we were student, and therefore all the students should utilize it. I just wanted to share while we were going through some data for the agri sector, particularly for the plant students who have interest in plant. May not be a student doing a stem cell research, can also be interested in doing plant. If you see the startup and all this area, just let me tell you, I was going through it. So if you see in Indian
30
agriculture,
seconds.
a very recent data has come which says that India has today 1,17,254 Indian startups. Can you imagine how many of those startups are from agri sector? Just guess. 80%. Okay, second chance. How much? 10%. Okay, 80 to 10. So now the real thing is it is only barely 3,000. Can you imagine? So just think how much is in front of you where you can contribute if you actually come up and do it, and it would be a largest growing sector in next 10 years, as Secretary DBT pointed out. And out of those 3,000, how many became unicorn? One more guess. It's only 100.
Okay.
So there is huge opportunity for the agri sector. So my purpose of standing here just to, tell you, if you are doing stem cell research, forget it. Come to agri sector and start working.
Maybe
Yeah.
we will partly disagree
For-
with Dr. Shubhra. We will totally disagree. Yeah. So, but what I want to really convey is thanks everyone. I
All the
think it's
best
been
for all of you for your future.
it's been really overwhelming, and I'm really glad the students asked some very brilliant questions, including the ones that have impact on policy. I also encourage all of you to get equally engaged in all your posters, and we have very mindfully separated the poster session into two halves. So the students who are not presenting the first half, please engage with your peers and discuss science, and vice versa. I also request the directors to not chit chat among ourselves, because we do that all the time, and there will be a lot more opportunities for us to do it. Please go and visit the posters. Two announcements. Since this is going to be one of the unique events, and like I said, it's going to be a momentous small event. We're going to be planting a tree where the secretary will of course plant it, but all the DBT directors will water it. Hopefully, it's symbolic. symbolic of the entire BRIC growth. Sangathan. I'm not very good in Hindi. The other thing is, of course, it's since you're in Cdfd, I would like all of you to also benefit from visiting our incubator facility, which has come up very nicely thanks to efforts from Rashna, Abhijit, Kavita, and others. If you have time, please do visit. It's an amazing facility where we're already incubating 6 companies and more are going to be in the offering. So thank you very much. We'll break for lunch and we'll see you after lunch. Thank you. There is a photo opportunity. Let's meet in front of the institution.
Yeah, CDFD may yeah, and there'll be lunch in the ground floor in the canteen. For the photo session, please come now.